I'm here. Hopefully it doesn't cut out. It's been a little bit sketchy the past couple days. I think I first saw you on Instagram, and I instantly knew that there was someone new on the scene. Someone was doing something special that I had not seen before. Actually, Stu, when did you start stacking? Was that the first way I met you?
Probably through the ID forum, but I started stacking like 3 years ago.
Yeah. But it quickly became clear that Stu was going to pay attention to things that other people weren't, and that he was going to do a really good job of imaging them. So it's really been my pleasure to get to know Stu in the past couple of years, not only through slime molds, but talking about other real, fungal sensu stricto organisms, not just slime molds. And most recently, Stu was on Santa Cruz Island with me and Julian, where he was taking small stuff, a lot of the fungi that I have no hope of making good images of myself. And so we really benefited from Stu's presence there last week, or whenever that was now, a week and a half ago. So I got so many requests from my students today to tell them anything at all about fungi, and I told them all to tune in. So hopefully some of my students are here tonight. And I will now turn it over to Stu to tell us about all the things that we don't normally get to hear about.
Thank you. Thank you, Dad, and everyone for having me. I'm gonna see if I can share. That's not right. Just a sec. Uh-oh.
Uh-oh. This is what we
should have tried this before. It's not letting me share my screen.
Oh, aren't you? You are co-hosted. Let me make sure everything is set up.
I think
you probably need to be made a co-host.
Yeah, he's co-host.
It's a computer setting thing. Let's try this. Okay, it's making me quit Zoom and reopen, so I'll be right back.
No problem.
Okay. There we go. Can you all see that?
Yep.
Mm-hmm. Okay. So yeah, thank you. I first got into mushrooms about 12 years ago. I was living in Portland, going to college. Started out hunting chanterelles, porcini on the Oregon coast. My interest was pretty much restricted to edibles for the first few years. I moved to Los Angeles after college for a job, and we had a pretty good rainy season shortly afterwards. And I realized, hey, I can kind of still look for mushrooms here too. But the next year was dry, it was a little bleak. Abandoned that. And around 2017, I started noticing slime molds when I was in Washington, visiting my family in Bellingham, Washington, where Jack and Lauren live. And at first, they were just kind of these beautiful alien little creatures that I saw when mushroom hunting. I liked photographing them with my phone, and soon the phone photos weren't good enough, so I kept upgrading cameras and flip-on lenses and did more and more with them, and I was kind of obsessed. I got back to LA and realized I could find them when there weren't any mushrooms. And I'll talk about why. But they kind of became a stand-in for my myco obsession because they were easier to find if you found the right microclimates. I'm not an expert. I don't have a biology or ecology background, so please feel free to correct me if I say anything stupid. I've just spent a lot of time looking for these things, observing them, photographing them, and would like to share them because I've noticed a lot more interest in slime molds despite them not being fungi in the mushroom community over the last few years. If you have questions, feel free to interrupt either out loud or in the chat. I'll try to keep an eye on the chat. Prefer to answer questions as they come up because it makes it more interesting. So don't hold back. And all of the photos in this presentation are my own, I think. If I see anything that's not, I'll point it out. And then I've borrowed a few diagrams that are not mine and tried to cite them. So what are slime molds? As I mentioned, they're not fungi. So bear with me, I'm still here for a reason. They're a class of amoeboid protists, which means that they're eukaryotes that are not plants, animals, or fungi that are mobile, and they change shape. So you have single nucleate cells that fuse into a macroscopically visible plasmodium, which you see here on the right. This kind of veiny, fanning dendritic structure that glides around logs or leaf litter, eating bacteria, fungi, and other microorganisms. And they've actually been characterized or classified as both animals and fungi at different stages, mostly in the 19th century. And that's partially because, like animals, they're locomotive, they move around. So early taxonomists, and let's call it an animal. And like fungi, they form spore-bearing structures that can really start to look like mushrooms. They're often on a stalk with a little ball on top that has spores, and they can easily be mistaken for mushrooms. Most of what I'm going to be showing you and talking about is really small, so even at the largest phase of their life cycle, most of the fruit bodies of slime molds are just 1 to 2 millimeters tall for most species. So on the left here, I have Trichiid Acipiens. I shot that photo with my phone. And then on the right, I used focus stacking, which Alan Rockefeller talked about in his talk last night, where you start out with the focus at the very front of the frame and increment it backwards until you get enough the frame in focus, and then stack those using third-party software. So these ones were probably about 2 millimeters tall. Just something to keep in mind about the scale as I continue to show some of my photos. One thing I really like about them is that they are kind of easy to personify. They form these really interesting little scenes. They're very charismatic and colorful. One of my favorite things when I'm walking through the forest looking for mushrooms is to flip a log or flip over a leaf and just see this bright flash of fluorescent color of slime mold sporangia. And I'm sure a lot of people here who spend a lot of time in the forest looking for mushrooms have had this experience. It's really just a surprise to see these really bright colors in nature like that when you're not expecting them. And Christian captured my joy in one of those moments last week on Santa Cruz Island when we went to a log that they had named the Stew log the day before I arrived. And I started pulling it apart and finding slime molds and tiny fungi and macroscopic bacteria that you can see. And I was just delighted. So here's a little phylogenetic diagram that shows where slime molds fall within the eukaryotic tree of life. Fungi and animals branched off a long time ago, but we are still talking about something that's more closely related to fungi. Hence, some of the similar characteristics, possibly. So they're not fungi. Why am I here at a virtual fungus fair talking about slime molds to a bunch of mushroom people? This is a photo of a Gymnopus, I think, with a Diderma species of slime mold that was fruiting on its gills. I think Christian found this last week and left it for me to photograph. the point is to illustrate the point that you'll find slime molds and fungi alongside each other pretty often. Whether you're a mushroom hunter, mycophilo, mycologist, whatever you want to call yourself, you've probably seen this happen at some point. And if you haven't, I hope you do soon. Another reason is almost every day on iNaturalist or one of the Mushroom ID forums on Facebook, I see slime molds misidentified as fungi, people asking for an identification, thinking it's a fungus, and I get it. I mean, why wouldn't you? It's right by a fungus, it's on a fungus, it has spores, it's weird, it's slimy, but they're not. But it just goes to show how much overlap there is in their habitats and their habits and the ways that people observe them. But ultimately, I think really why you should care is that they're really beautiful and fascinating organisms, and starting to take note of them and notice them will enrich your experience with whatever wild place you're spending time in, or even just in your neighborhood, in urban areas too. Not only through like their just sheer aesthetic beauty and their intricacy, but also because they kind of provide these glimpses into tiny worlds and teach us to start paying attention to things that we wouldn't otherwise normally. They provide really interesting insights into these microscopic worlds that include fungi. And just like fungi kind of were for me a gateway drug into biodiversity in general, slime molds have become another one, just taking me kind of deeper down the rabbit hole. So these are some of my favorite photos that I've taken recently, just showing the diversity of their forms and colors and structures and just kind of how weird and alien they are. And I'll get into more of what these are as I go on. So a little bit of biology first. I'm probably going to mess some of this up, but they start out as a spore, a haploid spore, which germinates, releasing like 1 to 4 myxomycetes or swarm cells that kind of glide around. They have a single nucleus. They glide around looking for mates, feeding, growing. At any point, either one of those types of cells can form a microcyst, which is kind of a dormant stage. It goes into a cryptozoic state that it can use to wait out for better conditions. So if things dry up, the conditions are not favorable for them to continue moving around and feeding. It'll form a microcyst, and then it'll be rehydrated later once the microclimate is more humid again, or it senses more food around, and it will wake up. Eventually, 2 or more of these cells will meet, and if they're compatible, they'll fuse. And once they fuse into one cell, the nuclei will fuse and form a zygote. And that cell, that nucleus, will undergo synchronous nuclear division, forming more and more nuclei within the same cell, growing and growing until you actually have a macroscopically visible plasmodial. So that's what I showed in that first stage. The slimy, which is kind of where they get their name, dendritic fanning structure that glides around in its trophic stage, eating for food, looking for food. Christian asked, do all Myxomycetes have the potential to form sclerotia? Yes. So as far as I know, they all have the possibility, the capability of forming sclerotia. So sclerotia is like a microcyst, but once they're a multicellular or multinucleate cell, they can form a sclerotium, which is another dormant stage. You can find these if you take the bark off of a tree, like during the dry months or the cold months, you'll see like little balls of, or amorphous globs of kind of, it almost looks plasticky, like yellow, orange, red. Plasmodium looking stuff. And these sclerotia kind of wait out again for better conditions to rehydrate, to sense food, and then they'll start crawling around as a plasmodium. Lauren asked, will I mention the differences of the amoeboid life cycle differences? So I think, as far as I understand, Lauren, both the plasmodial stage and the Myxomycetes stage are amoeboid, but I don't know too much more detail to, I think, answer the question you're asking. So eventually that plasmodium is going to turn itself into fruit bodies, sporulation phase. So it's going to reproduce. If it senses that nutrients have run out or conditions are drying up, in most cases, the entire plasmodium turns into fruit bodies. And you'll see in that top step K, they're forming stalks, little balls on top. The nuclei undergo meiosis and form the spores, which then mature inside the sporocarp, inside of a peridium that protects them while they're maturing. And eventually they disperse and the cycle starts over. So these are a couple of photos I took that I think capture at least the macroscopic stages of the life cycle pretty well. Maria said in the Myxomycetes, they only have one haploid nucleus, but in the Plasmodium, they have many diploid nuclei. That's correct. So you'll see on that last slide, it's separated in half between the diploid stage and the haploid stage. So basically, it's just sexual maturity and completeness of the chromosome. But I think the Myxomycetes and the Plasmodium operate very similarly, except for their scale. So these photos kind of show a few of those life cycle stages. This is Bodhamia utricularis. I found it on the outside of a log about 3 years ago when I first started stacking photos. It was first appeared, I noticed on the left, this scene after a few days of just light rain, and it was starting to dry up. The more translucent thin strand is more classic Plasmodium, connecting those two larger orange sections. And then you can see that more of the Plasmodium is starting to get more opaque. Some of it is crawling into that crack. You can tell that the environmental conditions are kind of worsening. The opaqueness is as it forms a sclerotium and dehydrates a little bit, but it's already turned some of itself into sporangium, which are the blue-gray balls over here. A few of them on the bottom are split open. I'm not sure if those are mature or they may have kind of aborted in the maturation process and split open because it dried out too fast. But I came back the next day, and on the right photo, you can see all of the mature sporangia, and the Plasmodium has crept into that crack in the log, kind of hiding from the UV exposure, the dry conditions on the outside, and it's become more opaque and more compact, and it's forming a sclerotium. So it's going to wait out for better conditions inside that log. And it didn't transform all of itself into sporangia, just enough to reproduce, and it'll wait out and come back out and start feeding. I never saw that one again. Typically, sporulation, the formation of fruit bodies, occurs on the order of about 24 hours. So if I find an immature slime mold, I'll try to come back 24 hours if I get the chance, 24 hours later, and check on it, because you typically need to see the mature fruit bodies to identify the slime mold. Usually the microscopic features are necessary in almost all cases. Fruit bodies are more frequently observed than the Plasmodium because in a lot of cases they come to the top of the substrate or somewhere more exposed where it's more efficient to disperse their spores, but not always. Then again, as I've mentioned, which is why it can be easier to find slime molds when you're desperate for fungi, microclimate conditions drive a lot of this behavior. So if you find a good wet log that's holding moisture well, and everything else is still dry around it, it's still worth looking. This is a time lapse that I took of Bauhemia nitens. So you'll see when I hit play, you'll see all around it, there's more Plasmodium kind of undulating, pulsing, looks like it's breathing. Some of it in the background, the larger orange or yellow spots in the background are going to become more mature sporangia like the one in the center of the frame, which is almost mature, not quite there. This species is pretty variable. It occurs on stalks sometimes, short stalks, sometimes it's sessile, no stalk, just directly on the substrate. But yes, you'll see. Is that still playing the video?
Yes.
The one in the center there is more mature. Once it's mature, it'll be more opaque, less slimy look to it. Then I think I took this home and about 12 hours later, I had mature fruit bodies. It loops a few times, but this was over the period of about 20 minutes. Oh, there we go. So I actually saw this species again last week. Maria found it on Ironwood, an ironwood log on Santa Cruz Island. And this was the most spectacular specimen I've seen of this species. Had these really beautiful yellow lime granules, calcium carbonate in the peridium, an iridescent blue layer underneath that, and like some orange in the bottom. Just really, really gorgeous specimen. Oh, my font color changed. It's a little hard to read. So a little bit about the anatomy of slime molds to hopefully give you a foundation of what to look for if you're curious about just looking for slime molds or identifying them. There are 4 types of fruit bodies that slime molds or Myxomycetes form. The most classic, which I've shown a few times so far, is the sporangium, which is typically on a stalk, sometimes sessile, on the substrate. It has these threads often inside called capillicium, which the spores adhere to, form on. The capillicium sometimes gives the sporangium structure. It sometimes actually assists in breaking open the sporangium so the spores are exposed and can be dispersed. At the bottom, sometimes there's this little cup called a colliculus that the capillicial threads attached to. An ethelium, so the most classic example of this is dog vomit slime mold, Fungus, which is the yellow one here in the top right. It's just a single larger spore mass. This is the exception to the rule. In most cases, where I said that slime molds are very small, I've seen ethelial slime molds get up to the size of about a baseball. In the case of Reticularia lycoperdon or Fungus, but it's all just one big spore mass. Morphodinia are all slime molds decomposers. I'll talk about that soon. Third type is a Plasmodial carp in the bottom left corner. This fruit body kind of maintains the shape of the Plasmodium more or less. it stays sessile on the substrate, but forms like little kind of sprawling sporangia without stalks. This is one that was not quite mature. I think it was Physarum cinereum on a eucalyptus twig in the same spot as that Bohemia nitens time-lapse. It's not about a year ago. But they kind of just form these sprawling maze-like plasmodial carpal fruit bodies. And then the fourth type is a pseudoethalium, which is a fused structure. It looks like an ethalium, like it's one single mass, but it's actually just fused sporangia. So this is Tubifera ferruginosa or something similar. Fairly common in the mountains here in the spring and summer. I guess more the spring. But it's a cosmopolitan species. So focusing on sporangia, I'll talk about the different parts of a sporangium. The hypothallus down at the bottom of the stalk is kind of like a remnant of the plasmodium that acts as a glue. It's often like a little disc around the base of the stalk that holds it to the substrate. Sometimes it's a sprawling kind of film across the substrate, and it really just keeps the sporangium stable so it can disperse spores there and not fall apart. The stalk, we call it a stalk, not a stipe like we do in mushrooms. The columella is an extension of the stalk that kind of extends into the spore mass. Not all sporangia have this. In those that do, the capolitial threads attach to the columella and extend from there and radiate outwards, giving the sporocarpid structure. D, the peridium. This is the outside layer right here. We also use this word when we're talking about truffles, like tubers. It's the outside layer. In some cases, it's made up of calcium carbonate. It protects the spores as they develop inside the spore mass and kind of just keeps it all together until it eventually gets broken off or falls apart and the spores are dispersed. The capolitium, those are the threads that the spores attach to and give it all structure. Some slime molds have a pseudocolumella, which is kind of just a short, stubby columella. You'll see this in like Diaderma species. Crabraria species have a peridial net, which is kind of like a birdcage-like structure in lieu of capolitium. It gives structure to the peridium, which then dissolves and the spores fall out through that net. And then the last one is the colliculus in the order Trichiales, like Arceria, Trichia. You have this little cup at the top of the stalk, the base of the spore mass, the capolitium attaches to. So here's another time lapse of Arceria denudata. This is, I flipped a log on a really dry and windy day and found this mature cotton candy slime mold that immediately started, the capolitium started expanding. I'll play it again. These ones have really an elastic capolitium that really expands and kind of pushes the peridium off when they're exposed to a change in like pressure or humidity. So it immediately started opening up. This is again like 15 minutes, and it kind of just expands and allows the spores to more easily get captured by air currents. Some studies have shown that slime molds with stalks are more efficient at spreading spores. Their spores are found over a greater distance because they lift them up off the substrate and expose the spore crop to more air currents. Then, do the spores originate from the capolitium, or is the latter just structural? In terms of what's happening inside, as each nucleus like decides what to be within the sporocarp, it's really complicated, and there's been some recent genomic studies that have shown that nuclei in different parts of the plasmodium kind of govern themselves despite being a single cell, and they all kind of decide what to become based on their proximity to other nuclei or external factors. So I think I'm not sure if the capillicium forms and gives rise to the spores, or if they just kind of occur alongside each other and adhere to each other and provide structure. Here's the same species. I'll start that again. Arceria denudata. I took this photo a few months later of a much nicer specimen. You can see that outer layer of the peridium starting to peel off. Capillicium expanding outside. This cup-like structure, the colliculus at the bottom, and some iridescence in the one on the left that's kind of caused by optical effects of light reflecting off of the spores and kind of getting refracted at different angles by the spores, or reflected at different angles by the spores back through the peridium and giving it that iridescent, like oil slick look to it. So there's lots of cool optical effects that occur. I'll talk about it more in a couple slides. Here's some micrographs that I took of spores and capillicium. On the left, we have, I think, a Ligonema schweinetzii. It's something in Trichiales. Really cool reticulation on the spores. Most slime mold spores, more or less, are on the same scale as most fungal spores. I'd say on average, they're about 10 microns in diameter. They are almost always spherical, excluding the ornamentation. So these ones on the left, they have this really cool reticulation. They almost look like polyhedral. And then the capillicium has these junctions and ornamentation like these rings around it. You kind of have to pay attention to when trying to identify. Then on the right side, we have Arceria ferruginosa, I believe. Arceria species are characterized by these ridges on their capillicium. These kind of like, looks like one of those ridge tubes or like an esophagus or something. You have to pay attention to like the anastomosing, the junctions, and the branching of the capillicium. And you can also see these little oil droplets, these green splashes inside the spores. You'll see that in fungi too. When you mount this on a slide, they kind of float to the top and you can see them at the top once you've prepared your slide. But most slime molds, as I said before, you really need to look at these features to identify them as species. So I'm always looking for mature sporangia to bring home from the field, or nearly mature sporangia that I can mature at home in a moist chamber, keep mostly intact. Here's more of the kind of optical effects I was talking about. So slime molds get some of their iridescence from chemical features, some from structural features, like I was mentioning, with the reflection and refraction through the peridium. This is a Laproderma species. I forget which. I identified it at some point. I saw this in Sequoia National Forest last summer. I photographed it using the focus stacking method that I mentioned earlier, which Alan Rockefeller covered last night. When you're shooting these really tiny things, you get your camera lens really close to them and you use extension tubes. And it's hard to light the whole subject, so I use an LED from below and a flash from above to kind of get this effect you see here. But Laproderma are one of my favorites. They're just always gorgeous. They're almost exclusively iridescent and have these cool mottled patterns and all these colors. So the ecology of slime molds is a relatively unexplored field. There are some studies that have been done over the last 30 or 40 years, but these studies can only focus on the macroscopic and visible stages of the life cycle. When they're myxomoebae or microcysts or spores, it's really hard to get a sense of what they're doing in their environment and how they're interacting with other organisms and what contribution those Myxomycetes make to detritus decomposition. So a lot of what I'm going to say is mostly observation and speculation, things that I've noticed, interesting tidbits that are worth exploring more. Slime molds are beautiful photography subjects on their own. They're really fascinating to shoot with a macro lens, but I like doing all of my photography, or almost all of it, in the field, because I like capturing these interactions with organisms and getting glimpses into these tiny worlds that you wouldn't otherwise really be paying attention to. So they, in their trophic plasmodial stage, they cruise around the substrate, eating bacteria, eating fungi, and they break stuff down, most importantly, releasing nitrogen and phosphorus back into the substrate. This is important because I think, as I think Jack Johnson mentioned on Tuesday, the decomposition of wood by fungi is very closely limited by the amount of available nitrogen in the substrate. So these slime molds, they will eat fungi, break down more complex molecules into nitrogen and phosphorus, releasing it back into the substrate. And this actually promotes fungal growth. So when you have fungi, you have more slime molds. And you can really find a really good log that has both. So we call them, I've heard them referred to as predators because they do move around and kind of hunt. But I think a better term is grazers, ecologically speaking. They kind of just, they're a certain kind of detritivore that grazes on detritus. What's interesting is that slime molds often appear more abundant during wetter and colder phases when soil microfauna like mites and springtails and stuff like that is less abundant. And mites also play a similar ecological role, releasing nitrogen back into the substrate. So it could be that slime molds are kind of taking over where the microfauna are less able to, and balancing that out. But the slime molds also provide a food source for the microfauna. In a few slides, I'll show a springtail that's actually eating spores. It's kind of just generally accepted that the more microscopic stages are playing an important role in the detrital food chain and nutrient recycling. But there's not a lot of quantitative evidence for it. There is some substrate specificity. Most slime mold species are not specific to a single type of like plant substrate, but they will prefer like either living trees or leaf litter or certain like textures of bark or like certain stages of decayed logs. But when it comes to sporulation and actually forming fruit bodies, they'll pretty much just form fruit bodies wherever it's most effective and beneficial to form them so that the spores can be dispersed. So in this photo, I found Laproderma scintillans on, I think it was a Chinese elm leaf. It might have been oleander. It was in urban Los Angeles in my neighborhood where I used to live. And it was actually fruiting, one of the sporangia was fruiting from a spider's head, or the molt of a spider's head. I like this quote from one of my textbooks. Although Myxomycetes distribution is almost undoubtedly limited by macroclimate and habitat, especially vegetation requirements of a particular species, there is strong evidence that microhabitat availability is the factor acting most strongly on species distribution. So this is the reason why you can find slime molds in so many places. And it's also the reason why a lot of slime mold species of the 1,000 that are described, or thereabouts, are cosmopolitan. meaning that they're found all over the world, or some in all temperate climates or all tropical climates. I have numerous times found a slime mold Myxomycetes species that's never been seen in North America, and it's just not really a surprise anymore. They kind of just pop up, and we need more people looking for them. But as I mentioned, sometimes slime molds and fungi occur alongside each other just because they like the same conditions. So this is Cryptomerasmus corberiensis is the mushroom. It's the olive leaf pinwheel, which Christian and Damon Ty have kind of been championing as a project to try to get one documented in every county in California. I'm not sure what we're up to with that yet, but I saw this one a few years ago in LA, and right next to it on another olive leaf was this Labraderma species. So as far as I know, they weren't interacting in any way. They just happen to like the same conditions for forming their fruit bodies. This is kind of an example of a really good log and how slime molds will indicate the presence of more fungi and vice versa, and how they have this mutualism. So the white slime mold is a Stematitis species, just starting to form. It's going to expand and get those chocolate tubes that you probably know. And then all around it are these Orbelia and Melissia or Melisiaceae. I don't think there's any way to tell what even genus those gray cups are. These ascomycetes. And it just kind of is an example of that perfect log habitat where there's just this mutualism. As I mentioned, some slime molds will eat fungi. So here we probably have Bothamia utricularis, which is a notorious polypore eater, the Plasmodium here. It's the same one I showed the different life cycle stages of earlier on the log. I often find this one on like Trametes versicolor or Trametes betulina. This is like some recipient polypore or some recipient porous crust or something. Then it was kind of crawling over and devouring either the fungus itself or whatever was on the fungus. Looks like there's some mold growing on it too, so it might have been secondary fungus. So they also interact in this way. And then one thing I've noticed is that I'm not sure if this is a case. I don't think it's coincidence, but I found this Comatrica nigra, the white sporangia, forming next to these gray Melissia cups. And I went back the next day and I noticed as I was shooting the slime mold again, there was a water droplet on one of the stalks that had a nematode in it. Right, Maria? I kind of just want to zoom down and walk around in this scene. So I think either Lauren or Jack mentioned that some fungi trap nematodes in little loops of hyphae and consume them for nutrients when they wouldn't otherwise get those nutrients. So in this next photo I'm going to show, inside this water droplet that was on the stalk of one of those sporangia, these blue streaks, it's a multiple exposure, it was stacked, and the nematode was kind of wriggling around. There was a nematode stuck in that water droplet. Presumably that droplet's going to fall and that nematode is going to fall prey to one of those Melissia, which I believe are known for having this capability of trapping nematodes. So just another possible interaction between fungi and slime molds to kind of keep an eye on. Interesting if it is true that they are interacting this way, but they do seem to have a lot of overlap in their ecological roles and their benefit to each other. This is what we call polycephalomyces tomatosus, or maybe something similar. Jack also talked about this in his talk on Tuesday. I think it's classified it's a fungus attacking the slime mold. I think it's classified in the order Hypocreales, which is also what cordyceps is in. So it's parasitizing this Trichia favigenea or Trichia scabra, something like that. The slime mold was way beyond identification at this stage. So there are these parasites. Here's another one. The slime molds in the top right corner are Cribraria cancellata. I forget what this mold is called. I figured it out at some point, but I forgot. But I frequently find it attacking these red, maroon, brown sporangia. And then on the left, the bottom left corner, you have Hemixomycetes candidus. It's a basidiomycete syphaloid fungus that I also often find inside logs looking for slime molds. And here's that same slime mold species, Cribraria cancellata. Really cool. These are about 1 millimeter tall. Thereabouts. There's some moss for scale. They have this cool birdcage-like structure, really pretty maroon, burgundy, sometimes purple colors, and they open up like this and the spores just fall out. There's also like associations with other insects and arthropods. So this beetle on the left is in the family Meodidae. It's I think they're called round fungus beetles. I see them eating Arceria and other Trichialia species pretty often. They really, really like them. They kind of just nestle and bathe in the slime or bathe in the spores. You'll see them like covered in spores. And then on the right is like a midge fly larva that I observed these things using their mandibles to cut open the like really tough calcium carbonate peridium of the Sphysarum species. And these things, I mean, it's like rock. When I crush them on a microscope slide, there's an audible crunch, and they're not easy to cut open. But these larvae were just kind of scissoring them open and eating spores like cereal out of the bowls. Presumably, the larva eats the spores. It matures into a fly with spores in its guts, and flies away, effectively increasing the range of spore dispersal for this species. So insects, arthropods, they're important vectors of spore dispersal for slime molds. Maria, great question. Are those protostellates on the larva, or are those just drops in the ends of the hairs? I had the same question when I first saw this. I kind of freaked out for a sec, and I thought, like, is this like some slime mold sporangium forming from the body of the larva? Like, is this a hyperparasite? But those are just drops. I got the fly identified at some point, and I got the answer that those are just kind of droplets, like insect exudate on the edges of the tips of the hairs. I wish it was the other option. I got lucky with this shot. This fly kind of crawled into the frame as I was stacking. This is a Meroderma species. It's a genus of slime mold that only occurs in snowmelt, which I'll talk about soon. I was just starting to use flash, a really fast recycle time flash for these photos. So the focus stack moved through the full depth of the fly fast enough because I had a really fast shutter speed, to get it without much blur. And it was in the same plane as the sporangia. So it was kind of cool to capture this. Same thing happened here. The springtail I didn't notice until after I started stacking this photo of. This is Arceria obvalata. It's a fairly common, probably in Santa Cruz, you have a lot of this. Fairly common slime mold. It's relatively large. They can get up to like 1 centimeter tall, and they expand when you pull up on a log, so they're easy to see. But the springtail was kind of just crawling around eating the spores. I stacked it fast enough that there wasn't too much blur. I like to imagine that it lives inside that little tunnel right there. And these photos came from Peter McDonald, who is a slime mold enthusiast and artist and teacher and researcher in Australia. She gave me permission to use these in my presentation. She found these eggs inside a Stematitis sporangium and kind of tracked them until they hatched. So apparently insects also use them to protect their eggs, lay eggs in them, and then also presumably this benefits the slime mold because the larvae end up eating the spores and dispersing them. So I was going to talk more about photography, but I think Alan talked about most of what I wanted to last night quite a bit, so I'll just give a little quick rundown. What you'll need to start finding slime molds. You'll want a jeweler's loupe or a magnifying glass for looking at really small things, and or a clip-on macro lens for your phone camera. Or if you want to go a little deeper, you can get a macro lens for your camera, extension tubes. There's a thing called a Raynox DCR 250. It's a 2.5 times magnification front lens. You can screw on the front of your lens. So between those components, I get up to around 5 times magnification when I'm shooting. I use an Olympus camera. They have built-in focus bracketing, so it'll actually do the series of shots, adjusting the focus for you, and you use software to stack it. And that's what most slime mold photographers use to get really nice, sharp, detailed images of small stuff. So one of the best spots to look is decaying logs, like we've discussed a couple times. There's kind of a Goldilocks zone. You don't want it too dry or not too wet. You might find logs in a creek bed or a stream bed. If they're too wet, you'll find like eyelash cups like Scutellaria on them, but not too many slime molds yet. But there's kind of, once you start getting the hang of it, you'll see that there's a sweet spot. You'll see a log and you're like, that's a good log right there. And you'll start taking it apart and find slime molds. Riparian areas are really good. I like canyons in my area that run east to west. So you have like north-facing slopes. They hold moisture really well. You can more easily find the microclimates you like. And then leaf litter after the rain. So alder leaves in the spring in the San Gabriel Mountains where I live. One of my favorite species is Diachia leucopodia. It's a beautiful blue iridescent slime mold with a white stalk. I'll show it to you soon. And then even like native plants or non-native plants, landscaping, invasives. Like I mentioned, like in LA, I used to find lots on English ivy, oleander, Chinese elm. You just kind of go out and look at the leaf litter shortly after the rain, like 12 hours after the rain. Things will start to kind of be at the stage where you can start to identify them and notice them. Here is a video. Oh, Dan asked, which Olympus camera do I use? I use the EM1. I don't have it right here. This is my old one with a microscope adapter attached to it. This is the EM5 Mark II. It's a mirrorless camera. They're really nice. They're really small, and you get a lot of bang for the size. It can really do a lot, and it's nice to carry this in the field because it's relatively small for what you're doing with it. But now I use the EM1 Mark 3. There's also the OM system. Olympus was bought by OM system. There's the OMD, which is similar. So here's a video of some really good slime mold habitat. Some of the things I found here I've shown, like the time lapse of the Bodhamia nitens. That yellow blob on the eucalyptus bark is the Bodhamia nitens. Some white stuff up there that's Serratia mixer particulosa, which I'll get into. It's not actually Myxomycetes. That's one of those Plasmodial carbs that I showed earlier. But that's kind of what you want to look for. Eucalyptus is really good. If you have like really wet, nasty eucalyptus debris in your neighborhood, now is a great time. Before maybe tomorrow, before the next storm begins, great time to go look at it and just pull it apart and dig it a little bit. When we were on Santa Cruz Island last week, we spent our last morning before we left just kind of digging through some eucalyptus debris outside the research station, and we found too much for me to photograph in the time we had. Another good spot is near other slime molds. So about half the time, if I find one slime mold species, if I keep looking at the same substrate, I'll find more. Smaller ones, different colored ones, different orders. They just seem to like the same conditions a lot of the time, despite not necessarily being very closely related. So if you spot one, keep an eye out. It's worth the time to kind of just keep digging. Here's another time lapse I did of a Physaurales Plasmodium kind of engulfing this Comatreca nigra sporangia. I don't think it's eating them. I think it's probably eating stuff on the substrate that's around them, but who knows? Maybe it's cannibalism. Or not cannibalism, they're pretty distantly related, but you know what I mean. But really my favorite spot to look for slime molds is the snowmelt. So the font color changed. I had Nivicolis slime molds and snowmelt slime molds down here. In the spring, I spent a lot of time in the mountains, kind of at the edge of the melting snow. As the forest floor begins to wake up, new stuff is decomposing. You have new organisms coming back to life. Reawaking after the winter, the slime mold Plasmodium starts to creep around, taking advantage of all this. And then as the snow recedes and the conditions dry up in the immediate patch of forest floor, they'll form fruit bunnies. So there's a lot of slime molds that have actually adapted to withstand colder temperatures and can only be found in these conditions. This one, unfortunately, was not one of them. This is an Arceria species, which is not Nivicolis Myxomycetes, but it just happened to take advantage of these conditions and fruit at that time. But you can see the sporangia kind of get deflated. They aborted in the middle of the maturation process and got a little too chilly. But I will show you some other Nivicolis species. This is me and Allison Pollack in Alaska last spring. If you don't know Allison, she's an incredible slime mold photographer, macro photographer who focuses on slime molds, but has really amazing fungi photography also. We went to visit our friend Bruce Walkovich in Anchorage, who hosted us and generously showed us around last spring. This little patch of forest in this video, we probably spent about 24 hours here over the course of 3 or 4 days. Just in this little area you can see in the video. And you can see little patches of snow. There were slime molds everywhere, from like salmonberry to Devil's club, so like live blueberry bushes that were still partially covered in snow. Um, just tons of slime molds everywhere. It's lucky that the days were very long up there in May in Alaska, because we needed many, many hours to just find them, let alone photograph them. This is a Lachnalula species on a fir twig, I think. Apes. In Western North America, we do have this phenomenon of snowmelt Nimicolis fungi, mushrooms, larger fungi that kind of take advantage of that same niche, and some really cool species. But with Nimicolis slimos, it actually occurs all around the world. Anywhere that gets covered in snow for more than 2 or 3 months over winter and then recedes, you'll see slime molds. And this fungus, the cup fungus here, this is not one of the snowmelt fungi that are specific to Western North America. This is Lachnalula, but just demonstrating how they kind of occur alongside each other in the snowmelt. This is a Meroderma species. This is the genus that is exclusive to snowmelt. They have these little funnels at the tips of their capillicium. So that's kind of like pockmarked peridium. Really neat. This is Lamproderma zonatum on, I think, some kind of, it might have been some kind of rabies thorns. Maybe it was salmonberry. But kind of cucumber-shaped, iridescent fruit bodies. They crawled on that spider. They kind of just fruit at higher elevations, relative elevations, wherever it's most beneficial to spread spores. And then that's me and Allison and Bruce again after a very successful trip. So I guess I was going to make this talk more about slime molds in California and spend more about slime molds in general. But the good news is most of what I've shown you can find in California. But these are some of the most common ones in California. You have Fuligo septica, wolf's milk, Lycogala, Neocarpus fragilis, Stemonitis, of course, Reticularia lycoperdon, that's a big epithelial slime mold that you'll find on like less decayed logs and sometimes in the big alder. Stemonitopsis typhida down here, they're like these hot dog on a stick looking slime molds. So I'll just show a couple of these so you can kind of know what to keep an eye out for. Reticularia lycoperdon, this one is huge. It's eaten in Mexico, I think in Oaxaca. We call it caca de luna or moon poo. I tasted this one, but it was probably too old. It was disgusting. It tasted like dirt and metal, which is unfortunate because it looked like chocolate on the inside. Lycogala epidendrum, one you've probably encountered, a lot of people have. They're kind of fun to pop. It's a slow-mo video. I took my phone. Be careful doing this. This stuff will stain your clothes. But when they're immature like this, you can pop them in. It's kind of like popping compulsive balloons.
Stu, you had a question earlier about if someone saw something they weren't sure if it was a fungus or a slime mold, what would they classify it on iNaturalist?
For California especially, just go ahead and call it a slime mold, and I'll hopefully see it at some point and correct you if it's not. But if you suspect it's a slime mold, it's probably going to get more attention on iNaturalist if you call it that, because there's a lot more fungi. And those of us looking at slime mold in California will eventually get to it. Thanks for bringing that up, Lauren. Was that Lauren? I think it was.
It was. You're welcome.
Trichidicipiens, salmon egg slime mold is the common name. I saw this one in Mendocino last spring or last winter about a year ago. It was covered in this spider web. It was so cool. It makes me wonder just how big these organisms are, how expansive these Plasmodia are. This fruiting of this slime mold covered an area probably about 20 yards in diameter. Just this huge area. It was all going off. It was everywhere. You would just walk down the trail for this 20 yards and everything was covered in orange dots. And I found this little patch. I was about to leave because I was driving home to LA that day. I was about to leave and saw this little patch that was just covered in spider web. And it was just so much fun to shoot the reflection of the slime mold in the droplets. I even caught one droplet that had a mite stuck inside of it and it was kind of all trapped in the spider's web. One of my favorite slime mold funds right here. Stemonitis species. On the left, that might be Stemonitis splendens, which is kind of what most Stemonitis gets called. An unnaturalist. On the right, this one's really common in my area. It's Stemonitis smithii. It has the smallest spores of any slime mold. And in that bottom photo, I actually blew the spores out. You can see the capolitial threads and kind of the internal structure a little bit more. And then just reshot the same photo with that. Stemonitopsis typhina. They kind of look like Stemonitis. They're in the same family. They're individual sporangia. They have this thin white film that engulfs the stalk and kind of turns into the hypothallus, with that glue around the base and the substrate. And that's the key to identify this species, which is fairly common. Here we have a little snail that was nestled inside a group of them. More of the Comatrica nigra that I showed earlier. This species undergoes a really cool color transformation from like white to pink to like red, then brown, then black, then eventually back to brown. And the spores are exposed. So this picture on the top right is kind of just the log was positioned so that it had different amounts of light exposure, and each stage of the fruiting was a different color, so you can kind of see the whole spectrum of colors at the same time. Arceria denudata, the cotton candy slime mold. They start out kind of white like this in the bottom right corner, and then look like cotton candy above that eventually. Arceria obulata, another one I mentioned. On the left, the background is Ceroporia spissa, like a recipient cephaloid fungus. You'll find like inside of logs in the spring. Bright orange. It made a really cool background for that shot. These are a good one to keep an eye out for. They're fairly large, easier to notice. I see them starting around this time of year, but more frequently in the spring. Crabraria species, these are the ones that have the perennial net instead of capolitium. I saw this up in Bellingham this summer. Bellingham, Washington, where Jack and Lauren live, is probably my favorite spot besides snowmelt that I've ever been to find slime molds. In the summer, especially if we get some rain, my family's up there, so I go pretty often. There's just tons of diversity. If you have some warm rain, these mixed forests, with like lots of Dunfir and alder and maple. There's just tons. So this is one of my favorite finds from last summer. Diachea leucopodia, this is the really pretty blue iridescent one that I see a lot of down here. It's super abundant. I recently saw it in December, which was really shocking, because it usually only comes out in like late March or April, and then tapers off around like the third week of May. But I found it fruiting in December, in this really rainy season last month. And here it is again on the edge of a bay laurel leaf. And here you can kind of see those optical effects where the reflection off the spores through the peridium is creating the iridescence. Serratia mixta. This is not a Myxomycete. It's in a related taxon called Protostelia. They form, you can kind of see it. It almost looks like cystidia on a really long cystidia on gill edges. They form spores on outside of the structures. Yes, if cystidia had a single spore on the end of them. But they form spores outside of the fruit body instead of inside. But they also are a good indicator for Myxomycetes. So even though they're not the same class, they do share some overlap in the niches that they inhabit. Another Serratia species, Serratia mixta. This is also in Bellingham. They form these really cool honeycomb-like structures. And then, can't read the text, I guess. Slime molds have kind of just taken me down the rabbit hole. So as I started noticing slime molds, I just went smaller and smaller. Started noticing things next to them, around them, on top of them. This was one of my favorite finds from Santa Cruz Island last week. It's a myxobacteria. The orange fruit body is on the right. And next to it is a Physarum species, which is, each one of those little globes is probably about 1 millimeter in diameter. So the myxobacteria is very, very tiny, like 0.2, 0.3 millimeters tall. Myxobacteria have a similar life cycle to slime molds. They cruise around as swarms of cells instead of single-celled plasmodia. Then they form these sporangia, or I forget what the term for them is. Similar to slime molds. So just noticing these things has kind of opened my eyes to all these different organisms and things that I never would have noticed before, and really just expanded my interests in biodiversity in general. Here's the myxobacteria again. I had to crop it quite a bit, so it's grainy. And then as I've gone deeper, the slime molds have kind of just led me back to fungi. So here's our Syrian denudata again. And something that I would have called a zygomycete, but I guess zygomycota is broken up, so I'm not sure what to call this anymore. But just these little fungal threads that you start to notice next to slime molds, and get smaller and smaller until you're just back where you started, interested in fungi again, and you're looking for mushrooms again, which is kind of what happened this year because it's been such a good mushroom season. So I'll finish with this picture of a Physarum species on a tan oak leaf that kind of led me, by noticing these little yellow splotches, I found a patch of candy caps that this was resting on. And wouldn't have noticed the candy caps if it weren't for the slime mold. That's kind of all I got. Some resources, some social media accounts to follow if you like these photos. Allison, I mentioned Allison. Sarah Lloyd is kind of the OG slime mold influencer on Instagram. She's in Tasmania. She makes a lot of really cool ecological observations. Talks about them in her posts. Posts great photos of species that are endemic to Australia or that region. Leah Benlin started the Slime Mold Identification and Appreciation Forum on Facebook, which is a great resource. If you want to identify slime molds, you can post them there and we'll try to identify them for you. Barry Webb is an incredible photographer. Takes lots of great slime mold photos. And my Instagram is Sir Mix-a-Lot. And if you have any questions, you can email me. My email's right there. Books, Myxomycetes by Steve Stevenson. It's out of print now, but I think there's still copies floating around. There was a new edition a few years ago. It's a good short little intro. Gives you the basics, some common species, tells you what to start looking out for. Sarah Lloyd has a couple books she's published with the slime mold creeps. If you contact her on Instagram, you can ask her how to purchase her books right now. She might not have them right now, but she does infrequent releases. This is a really good one for our area, A Guide to Temperate Myxomycetes, and it's still in print by Nananga Burmakamp. It has some good keys in it, no photos, pretty decent illustrations. That's the one that I would most immediately recommend as a field guide. That's still in print. And then the Myxomycetes by Poulain et al. Is a 2-volume set, 2 gigantic volumes. One is all keys in French in the first half of the book, English in the second half of the book, and then just every single photo, any slime mold you can think of. Almost all described species, there's photos of them, diagrams of their spores. They're very expensive now, they're out of print, but you can probably, hopefully find them on eBay at some point if you really, really want to invest in this. And then I misspelled that at the bottom and didn't finish typing it, but there's a textbook by edited by Carlos Rojas and Steve Stevenson, Myxomycetes. It's more of a technical book, has lots of genomics and ecology, less of a field guide. And that's all I got. Any questions? No more? All right, thank you all. I'll give it back to Dan.
Oh, I'm just checking to see if there is any final questions on the YouTube chat group or on our chats.
Vanessa asked, what type of camera or lens would you recommend for viewing slime molds? Olympus. Christian, I think you use a TG, right? Damon Ty uses a TG. The Olympus Tough TG series is really good. If you don't want to really invest in a full-on mirrorless camera and the whole setup for focus stacking, TG series cameras do a pretty good job starting out. I know Alan uses a Nikon, which has focus stacking built in. The thing I like about Olympus is that when you do the focus bracketing, you can actually see on the viewfinder each photo that it takes. You can stop it whenever you've taken the focus far enough back. On the Nikon, you can't, the screen goes black. You can't tell how many photos or like how far back in the focus in the field you are yet. So you don't know when to stop, and you kind of just have to guess. And get the hang of how many photos you think you want to take. But with mine, I just set my camera to like 300 photos, and then I just stop it once I have everything in focus that I want. Chloe made the good point that the Olympus TG6 is also waterproof, so it's really great for tide pooling, if you like that.
Yeah, I use a TG4 and it's good for snapshots like my background photo. It only stacks 10 photos. So you're a little bit limited, but you can choose the depth to some degree.
I think it still does a really good job of getting important features, and I'm thinking about getting one myself, just mostly for the waterproofness. And it does a good job. I mean, I want something that's faster to document stuff with.
Yeah, and it's you know, it has good enough image stabilization for a little camera that you can do stuff handheld in pretty low light. And you know, I use a ring flash that well, actually it's not a flash, it's actually a light guide for the LED.
Yeah, and that's made by Olympus too, right? It kind of comes in the package. Yeah, they have some nice accessories for it, so I recommend that if you want to look for a used TG4 on eBay or some used camera website, if you want to start out and decide if you want to invest more. Were there any YouTube questions?
No, I think that's covered it. We've got about 15 people lurking there.
Stu, where can we get lay Myxomycetes? Is it impossible?
It's took me like a year to get it. It's really hard to find.
So we have to be very committed and check eBay compulsively.
Yes, like set Google alerts, and I went up to Portland last summer, I think, or like 2 summers ago, and Adam Bryant, if you know Adam, I met him for the first time, and he was like, you know what, you're gonna use this book more than me, and he sold me his set, and I had been unable to find it, and he had bought it a few years before when it was still in print. But it's a really incredible book. I mean, it has photos of every single species for the most part that's been described, and they're good photos, and the keys work great. It's my favorite book, I think.
That's so nice of him to sell you, because
I'm lucky to have it. But yeah, keep an eye out. It's, even as a casual slime mold appreciator, it's just a beautiful book to have.
I see Uncle Don has a comment. Well, I'll let you read it.
Thank you. I gleaned some more info on how slime molds fit into a forest ecosystem. I suspect they are facultative, beneficial, if not obligate partners to fungi and other organisms. I think you're probably right. They might be obligate. I think a lot of, especially saprobic fungi, wouldn't exist if it weren't for slime molds, which are a more ancient lineage. They probably set the stage in terms of nutrient cycling to make the evolution of fungi possible. But that's just speculation. I don't know much. A lot of sources talk about them as peripheral in ecosystems. I think that's an oversight. It's just that they're hard to observe and really understand their ecological importance, and they're really cool. And seeing these little tiny worlds just makes you more and more aware of all these things that we're not thinking about ecologically, as Jack talked about in his talk also. Gordon asked, does Plasmodial excrete waste? Yes, I've been asked this question before and I researched it. I forget the mechanism, but you'll often see like slime mold tracks on certain substrates. These little black, they look like Plasmodial, like ghosts of Plasmodial, where it used to be. And I have a few sticks where there's like permanently imprinted tracks of slime mold waste on them. and you can see the evidence they were there. But Gordon, I forget the mechanism by which they excrete waste. Thanks, Lauren. I'll be back soon, probably in the summer, to find more slime molds. Gordon also asked, I know slime molds engulf stuff to eat it, but it seems like they must release a bunch of enzymes externally too. Yeah, I think that's what's going on. They have like, whatever they're excreting is pigmented and it stains the substrate. I'm not sure what it is, but maybe that's, I mean, it's probably where the nitrogen and phosphorus it's getting back, added back to the substrate is coming from. All right. Thanks for having me, and thanks everyone for coming.
Any more questions? I guess I should say, yeah, be sure to tune in tomorrow night for, well, we were just having a local mycologist, well, semi-local, Christian Schwartz giving an update on the sea islands, sky islands, and dry islands.
Can't wait for that.
So I guess with that, I'll adjourn. People can hang around and chat as long as they want. I'm going to stop recording and